Hello, this is my DNA replication video, and first we're going to talk about the role of helicase, which is seen as D right here. And what helicase does is it breaks the hydrogen bonds between the nucleotides in the DNA and allows it to split up as it does. And next we have topomerase, which is seen as C right here, and relieves the stress on the whole DNA when it is unwound and split up. Next we have DNA polymerase 3, which is seen right here. And something to know is DNA polymerase 3 can only work in a 5 to 3, from 5 to 3. Remember that, we'll cover that in a bit. And DNA polymerase 3, what it does is when activated by these primers, it pretty much it grabs DNA nucleotides and places it onto these strands and just continues down the line. Okay? When it's working from 5 to 3. Next, we have DNA polymerase 1, which is seen as B right here. And what it does is that it replaces these primers with DNA nucleotides. Okay. Next, we have ligase, which is seen as, I believe, A right here. And what ligase does is that since between every Ozaki fragment, which we will discuss in a bit, there's these little gaps, and what ligase does is that it fills in those gaps with DNA nucleotides. Okay? And what does semi-conservative mean? Semi-conservative means that it keeps half of the original, which is seen here, because it keeps half, there's two pieces of the original in both of these segments. And what's the difference between the lagging and leading strand? So the difference between the lagging and leading strand is that the leading strand goes from 3 to 5, okay? And the leading strand is able to be done continuously. Since it is anti-parallel, the opposite to the 3 would be the 5. So it would be going to a 5 to 3 for DNA polymers, and it would be able to work continuously down the line. However, in the lagging strand, we see that it is a 5 to 3, which means that the anti-parallel, since it is all anti-parallel, the DNA polymers cannot work since it is going from 3 to 5, not 5 to 3. Okay? So what has to be done in the lagging strand is multiple primers have to be placed down, and then the DNA polymers has to go back and then just work down, then hop to the next primer, then work down. This leads us to our next thing, which is Ozaki fragments. These small sections where the DNA polymers are going all the way down. It's going all the way down. Makes these little segments. They're not all filled in. There's little gaps between the primers and the actual DNA nucleotides. And these are all called Ozaki fragments.